Hello everybody and welcome to Bash Course Quarantine Biology. My name is Faith and today we are going to talk about the cell cycle. So I said this video was going to be about the cell cycle, and it is, but that's going to be a broad title including cell division, both mitosis and meiosis, the cell cycle control system, and even the development of cancer. That being said, we're going to begin with cell division. Cell division is a defining part of life, but it is simply how two daughter cells are made from one parent cell. This can be the same as reproduction, the production of new organisms, if you are a unicellular species, but it's not the same for multicellular species. There are two types of reproduction. Asexual reproduction only involves one parent and includes binary fission, budding, and fragmentation. Sexual reproduction involves an egg being fertilized by a sperm, and so requires two parents. It makes use of gametes, each containing half the genome. Because the offspring will have a unique combination of genes, they will be non-identical to their parents, and this is increasing the genetic variation of this approach. Okay, so now let's talk about the cell cycle. This is the sequence of events in a cell's life as it goes from its formation to when it's going to replicate. It's divided into two main regions: interphase, the phase of growth and development and normal cell functions, and the M or mitotic phase, which is when the cell is going to divide. Within interphase, there is the G1 or gap1 phase, which is growth, the S or synthesis phase, which is DNA replication, and the G2 or gap2 phase, which is more growth. Within the M phase, there's mitosis followed by cytokinesis, and that's what we're going to talk about next. So mitosis has four main phases, 5 if you subdivide the first one, prophase, into prometaphase as well. In prophase, the chromatin that was duplicated during the S phase condenses into chromosomes. Each chromosome is composed of two sister chromatids attached at a centromere. Also, the mitotic spindle is going to begin forming from the centrosomes. Those are regions of the cytoplasm which contain the centrioles, and they're also known as microtubule organizing centers. In prometaphase, the nuclear envelope is going to break down. The spindles are going to reach the chromosomes and attach at their centromeres to the kinetochore proteins. Proteins are then going to begin moving the chromosomes to the center of the cell. In metaphase, the spindle is going to be fully formed. It's like a football-shaped thing of microtubules, and the chromosomes are going to line up at the metaphase plate. In anaphase, the two centromeres are going to be pulled apart as the chromosomes are moved to opposite poles of the cell, and the cell is going to lengthen. In telophase, the two daughter nuclei are going to begin to form as the chromosomes are going to decondense from the chromosome form into chromatin, and the spindle is going to disappear. Finally, in cytokinesis, the cytoplasm divides and the two cells separate. However, cytokinesis happens differently in plants and animals. In animals, you're going to see a cleavage furrow as a microfilament ring contracts. And as that furrow is going to deepen and the ring contracts more, eventually it's going to split the cytoplasm of the two cells apart. However, in plants, you're going to have vesicles containing cell wall materials, and they are going to kind of line up in the center of the cell and then fuse into cell plates. These plates are going to expand until their membranes hit the plasma membrane, and then the contents of the vesicles, the cell wall materials, are going to join with the existing cell wall and split the plant cell, into two new cells. So there are several types of conditions that are going to stop cell division from happening. These include density-dependent inhibition, which also includes contact inhibition, which is where proteins of one cell interacting with proteins of another cell are going to create a signaling cascade that's going to tell the cell not to divide anymore. And anchorage inhibition, where cells are going to require a solid surface in order to be able to keep dividing. So we've talked about mitosis. Let's move on to its companion, meiosis. This is the process that forms gametes, which means it must somehow cut the amount of genetic material in half. For humans, this means reducing the number of chromosomes from 46, the diploid number, to 23, the haploid number. Our somatic or body cells have 44 autosomes and 2 sex chromosomes, but they are paired up by which carry the same genes into sets of homologous chromosomes or homologs. Males obviously have non-identical sex chromosomes, however, being XY. Meiosis accomplishes this by going through 2 rounds of phases, meiosis 1 and meiosis 2. In meiosis 1, things happen just like in mitosis, but with a couple of key differences. In prophase 1, when the duplicated chromosomes condense, they're going to line up in tetrads, which are pairs of homologous chromosomes. This is called synapsis. And parts of their arms are going to be exchanged through a process called crossing over. The junction points are called chiasmata. At this point, the cell is still 2N, which is the abbreviation for diploid, even though its genetic material is duplicated. In metaphase 1, the tetrads are going to line up at the metaphase plate, and in anaphase 1, they're going to be pulled apart. After telophase 1 and cytokinesis, the two daughter cells are each considered haploid because they each only carry half the genes, even though the genes they do carry are still duplicated. The sister chromatids are still attached. Meiosis 2 is going to take care of that, with each of the daughter cells having their sister chromatids separated and split into two cells, so that in the end there are 4 daughter cells, each haploid but unduplicated. Sometimes things can go wrong in meiosis, and usually we can tell what has happened by looking at a karyotype, a photograph of the chromosomes in pairs all lined up. Non-disjunction is an error that occurs when a pair of chromosomes don't separate, but this can happen in meiosis 1 or meiosis 2. The results of this are various disorders, such as Down syndrome, also known as trisomy 21, where non-disjunction causes an extra chromosome 21. There are also many disorders due to non-disjunction of the sex chromosomes, such as Klinefelter syndrome, which is XXY, and Turner syndrome, where you only have one X chromosome. Sometimes, if gametes end up 2N instead of N, for example, the zygote can end up with 4N or more. This is called polyploidy and can even lead to the formation of new species. This whole process of cell division is regulated by something called the cell cycle control system. This is a group of molecules that kind of regulate and coordinate the cell cycle, and it involves multiple checkpoints that are going to trigger the separate phases. If the system does not receive like a go signal at one of these checkpoints, the cycle is going to halt. This depends on both internal and external signals and signal transduction pathways that utilize both kinases and phosphораses, which phosphorylate and dephosphorylate molecules in a signal transduction pathway, respectively. There is a G1, a G2, and an M phase checkpoint. However, if a growth factor does not get to the G1 checkpoint, the cell will enter the G0 phase. This is like fully developed nerve and muscle cells, and that's where the cell is no longer going to be dividing. And cancer can occur due to alterations in the cell cycle, because that'll cause the uncontrolled growth of cells that we associate with cancer. Also, changes in the chromosome structure can lead to disorders as well as cancer, and these changes include deletions, duplications, inversions, and translocations. So that's all of the cell cycle, and good luck. Thank you all for watching Bash Course Quarantine Biology, and I'll see you again next time. The Bash Course Quarantine team is Sandro Ansari, Sam Mencimer, Eamon Cole, Pasha Ishaq, Sarah Orozco, and myself. Please put any questions you have in the comments below, and we'll try to answer them or get Dr. Green too. Remember to tune in next time on Bash Course Quarantined, and as they say in my hometown, oh my EOSH.